Good morning, students. We are discuss about anatomical structure, right? Tissue system in the dicotyledons and the monocotyledons plants parts. In earlier lecture, we have learned about primary structure of dicotyledons root and the monocotyledons root, and what is the difference between monocotyledons and the dicotyledons root, right? So all the detailing study about monocots and the dicots root we are learned in a previous lecture, right? Now in this lecture we are discuss about anatomical structure of dicotyledon stem and monocotyledon stem, and what is the difference between in the monocotyledons and the dicotyledon stems. So let's start. Same as a root, similar in a stem, a similar system of the root, right? So internal structure of the dicotyledon stem, and this image is a sunflower stem, right? And the systems are the differentiated in three regions, right? Dermal regions, ground regions, and the vascular regions. So also they are the differentiate in epidermis, cortex, endodermis, pericycles, xylem, and the phloems. All the parts are the present in the dicotyledon stems. So here the represent of both image. One is a schematic diagrams of the dicotyledon stem, and another is a detailing structure of the dicotyledon stem, and these both are the transverse sections, right? So let's start. First one is epidermis. Epidermis is the outermost layer, right? They are the single layer, and their main function is a protective layer, right? And epidermis are the normally single layer parenchymatous cells and rectangular cells, right? Cell shape is a rectangular, and the cells are the compactly arrangement. Here no any intercellular space are the presence. The outer walls of the epidermal cells have a layer called is a cuticles. In a stem also cuticles layers are the present, and the cuticles is made up of the waxy substance, and it's known as a cutin. Cutins નું એક layers હશે stem ની upper side, right? એટલે કે epidermis ની upper side, એટલે એ protective layer બની જાય. So stomata may be present in the epidermal layer. In a stem, some cases have a stomata. Normally stomata is present on the leaf, right? But some species of the dicotyledon plants and the monocotyledon plant having a stomata. Epidermal cells are the living cells, right? And also epidermis are the made up of the parenchyma, and parenchyma cells is a living cell. So that's why the epidermal cells are the living, and the chloroplasts are the usually absent, right? Chloroplasts are the usually absent, and the large number of the multicellular heads occurs on the epidermis. In a root system, the root hair is a normally unicellular, but in a stem the root hairs is a multicellular, right? The next region is a cortex region, and this is a very broadest region in this case. In this case that's when the internal structure, right? And internal structure in the cortex no region છે મોટો જ હોય છે. So cortex lies below the epidermis, and the cortex are the differentiated into three regions, right? So below the epidermis there are the few layer of the collenchyma cells, and this zone is called the hypodermis. એક્ઝેક્ટલી કોર્ટેક્સની નીચે 2 થી 4 જેટલા લેયર્સ હોય છે, જે લેયર્સને હાઇપોડર્મિસ કહેવામાં આવે છે. And it gives the mechanical strength to the stem. And these cells are the living and thickened at the corners. Corner બાજુથી એ thickening હોય છે, thick હશે. And inner to the hypodermis, a few layer of the chlorenchyma cells. Chlorenchyma, that means chloroplast is present, and with conspicuous intracellular space. These chlorenchyma cells are the loosely arranged, so that's why intercellular space are present. And this region performs the photosynthesis, and some resin ducts also occur here. અમુક કેસની અંદર આપણને ખ્યાલ છે કે સ્ટેમ છે પોતે પણ ફોટોસિન્થેસિસની અંદર પાર્ટિસિપેટ કરતું હોય. તો જે પેરેન્કાઇમેટસ સેલ્સની અંદર આપણને ખ્યાલ છે જે ક્લોરેન્કાઇમેટસ ટાઈપના જે પેરેન્કાઇમા છે, રાઈટ, સો દે હેવ અ ક્લોરોપ્લાસ્ટ. તો જે ક્લોરોપ્લાસ્ટ હશે તો ફોટોસિન્થેટિકની અંદર એ પાર્ટિસિપેટ કરશે, પ્રોસેસની અંદર પાર્ટિસિપેટ કરે છે. The third zone is made up of the parenchyma cells, and these cells store food material. So in a dicotyledon stem, and especially in sunflower, the cortex is differentiated into 3 regions. The cortex is differentiated in 3 regions. First one is lies below the epidermis, and it's called hypodermis. Hypodermis layers are more than 2 to 4, that means a few layers of the hypodermis, right, and they are the very thickened, right. And the next one, some layers after below the hypodermis, few layers of the chlorenchyma. Chlorenchyma, that means having the chloroplast, and the last one is a few layers of the parenchymatous cells, and they are the store the food materials, right. Then after the cortex, single layers is present, and they called endodermis, and it's the innermost layer of the cortex. Cortex ની નીચે આવે છે એટલે innermost કીધું, because cortex, hypodermis and endodermis, they are the ground tissue, so that's why they are the innermost layer, right. The cells of this layer are the barrel shaped and arranged compactly without intercellular space. Here also endodermis are the very compactly arranged, so no any intercellular space are the present, and since the starch grains are the abundant in these cells, right, and this layer is called also known as a starch sheath, because the starch grains are the present, so that's why the regions is called starch sheath. And in most of the dicot plant, dicot stems having the endodermis with Casparian strip is not developed, and the central part of the stems inner to the endodermis is known as to stele, right. After the endodermis region is called stele, and in the stele, xylem and the phloems both are arranged in the different manners, and the xylem and the phloems surrounding one single layer, and this called the pericycles. So it is occur between the endodermis and the vascular bundle, right. Pericycles is surrounded by the xylems and the phloem, that means the vascular bundles, clear. So in the stem of the sunflower, pericycle is a heterogeneous cells made up of the both like the parenchymatous cells and the sclerenchymatous cells. Normally pericycles are the made up of the homogeneous cells, but in this case, in this plant and some another plants having these pericycles are the heterogeneous in the nature. Heterogeneous, that means different kinds of the cells through making this layer, making this ring. Like this pericycles is normally making by parenchymatous cells and also sclerenchymatous cells. So that's why the regions are the heterogeneous cells, right? Heterogeneous layer. Then the next one is a vascular bundle. The vascular bundles consist of the xylems and the phloems, and they both rings, both bundles are the differentiated by the cambium ring. And the xylems and the phloems in the stem occur together and they form the vascular bundle. જો એકબીજા સાથે ભેગા થઈ જાય તો એને આપણે શું કહીએ? vascular bundle. The vascular bundles are the wedge shape and they are arranged in the form of the ring, and the vascular bundles are the different kinds of the arranged, like the conjoint, collaterals, open, endarch, any kinds of the arrangement. Next, in the vascular bundles separately characteristics about the phloem. So primary phloem lies toward the pericycles, right? And also we are the study about the primary structure of the dicotyledon stem. So here now we are the study about the secondary growth of the plants. So obviously primary phloems are the near to the pericycles because xylem surrounded by the phloems. એટલે કે xylems is placed in the inner side and the phloem is the outer side. So in this case we are the compare to the distance, so xylems and the phloems. So phloems are the very near to the pericycles, and it consists of the protophloems and the metaphloem. જેવી રીતના પ્રોટોઝાયલમ્સ મેટાઝાયલમ છે એવી જ રીતે પ્રોટોફ્લોએમ્સ એન્ડ ધ મેટાફ્લોએમ્સ. Phloems consist of the sieve tubes, companion cells and the phloem parenchyma. So here phloem fibers are the absolutely absent in the primary phloem, but after this growth, that means the secondary growth having the phloem fibers, right? Then cambium rings. So their cambiums consist of the brick shape. ઈંટની જે એરેન્જમેન્ટ હશે ઈંટ જેવી રચનામાં જોવા મળશે. બાજુની ઇમેજમાં યલો કલરનું લેયર છે તમે જોઈ શકો છો, અને યલો કલરનું જે લેયર છે તમે જો ઉપર બ્લુ કલર છે એ છે ફ્લોએમ છે. દેન આફ્ટર ધ રાઉન્ડ શેપની અંદર તમને એરેન્જમેન્ટમાં જોવા મળે છે એ ઝાયલમ છે. તો બંનેની વચ્ચે આ રિંગ છે, રાઈટ. ઇટ ઇઝ 2 to 3 layers in the thickness. 2 થી 3 લેયરની અંદર એની થિકનિંગ છે એ આપણને જોવા મળે છે. These cells are the capable of forming the new cells during the secondary growth, because the cells is a meristematic cells, and the meristematic cells having the differentiating capability, right? Meristematic cell છે એની અંદર શું થાય છે? ડિફરન્શિએટ થવાની કેપેબિલિટી હોય છે, ડિવિઝન્સ થશે. એટલા માટે કેમ્બિયમની રિંગમાંથી જ નવા નવા રિંગ ફોર્મેશન છે, સેકન્ડરી ગ્રોથ છે એ થશે. Then after the xylems, so xylems consist of the xylem fibers, xylem parenchyma, xylem tracheids and the xylem vessels. So all the parts are the present, right? And the xylems is a conduct of water and the minerals from the root and transport the all over the plants. So here another one region, another one ring is formed, and is called a medullary rays, right? So few layer of the big polygonal shape, and lying in between the two vascular bundles are the medullary rays. બે બંડલ જે હશે વાસ્ક્યુલર બંડલ એની વચ્ચે આપણને પટ્ટા જોવા જેવી રચના જોવા મળતી હોય, લેટરલ, રાઈટ. લોન્જિટ્યુડનલ ટાઈપ. તો એ બેયને સેપરેટ કરતા એવી એક રિંગ ફોર્મ જોવા મળે છે જેને આપણે મેડ્યુલરી રેસ તરીકે ઓળખીએ છીએ. So this store the foods and the water materials and also function in the internal conductions. Then the last one is a pith. So in a large central portion of the stem is called a pith. આપણને આની અંદર પીથ છે એ જોવા મળશે કારણ કે ઝાયલમ ફ્લોએમની એરેન્જમેન્ટ છે એ સેપરેટ બંડલની અંદર જોવા મળે છે, જે તમે ઇમેજમાં જોઈ શકો છો. એક બંડલ નથી, ઝાયલમ ફ્લોએમના આવા ઘણા બધા બંડલ હશે, રાઈટ. અને આવી રીતે સેપરેટ બધાની અંદર એન્ડોડર્મિસ, પેરીસાયકલ્સ આ રીતની રચના પણ હોઈ શકે છે, રાઈટ. સો એટલા માટે પીચ છે એ આની અંદર આપણને લાર્જ રિજિયનની અંદર જોવા મળશે. So it is a composed of the parenchymatous cells with intercellular space because they are arranged very loosely. The pith is also called known as a medulla. Pith ને બીજા કયા નામે ઓળખવામાં આવે છે? Medulla. And function of the pith is a storage of the food materials. Right, so these all the informations about a dicotyledon stem. Now we are the study about a monocotyledon stem. So what is the difference? લાસ્ટ માં જોઈએ ડિફરન્સ શું છે, રાઈટ. એ તો જોતા ખબર પડી જશે, ઇમેજ જોવો છો એના ઉપરથી પણ તમને આઈડિયા આવી જશે. So normally outlines of the monocotyledon stem and we are seeing the maize stem, Gia maize stem. So in the transverse sections is a more or the less circular, and they are the differentiated of the epidermis, hypodermis, ground tissue and the vascular bundles. So monocotyledon internal structures are the differentiated in this region. First epidermis, then next one is the hypodermis, then the ground tissue and the vascular bundles. So in this image all the detailing structure we are the observe. So outer layer is a epidermis, and yes, obviously epidermis is made up of the parenchymatous cells, and they are the single layer. The outer walls are the covered with thick cuticles, right. So cuticles નું layer હશે. આના આગળના કેસમાં જોયું જેમાં એપિડર્મિસ ઉપર ક્યુટાઇન્સ નું લેયર હતું. And the continuity of the thin layer may be broken here and there by the presence of the few stomata, and multicellular hairs are the absent. In this case, in a monocotyledon plants having a no multicellular stomata, otherwise the present of the hairs, so the hair is a unicellular hairs, not a multicellular. And another case, hairs are the absent. Then next one is a hypodermis layer, and the hypodermis layer is lies below the epidermis, and some layers of the hypodermis, like a few layers of the sclerenchymatous cells lying below the epidermis, constitute the hypodermis. That means the hypodermis is made up of the sclerenchymatous cells, and they are the usually 2 to 3 layers, right. And these layers give the mechanical strength to the plants. And then next one is a ground tissue, that means a cortex region. So there is a no distinct into cortex, endodermis and the pericycles and the pith. So here ground tissue are the not differentiated in these layers. તો એકને જ આખે આખું ground tissue કહેવાશે, right. So the entire mass of the parenchymatous cells lying inner to the hypodermis forms the ground tissue. Ground tissue છે એ ક્યાં જોવા મળશે? એપિડર્મિસના નીચેનો આખે આખો ભાગ છે એ પેરેન્કાઇમેટસ સેલથી ઘેરાયેલો હશે. તો હવે જે પણ લેયર હશે, એન્ડોડર્મિસ, પેરીસાયકલ, વાસ્ક્યુલર બંડલ, એ સેપરેટલી એમ્બેડેડ થયેલા હશે ગ્રાઉન્ડ ટિશ્યુની અંદર. જે તમે જોઈ શકો છો અલગ અલગ બંડલ છે, અલગ અલગ એનું એન્ડોડર્મિસ હશે, અલગ અલગ એની પેરીસાયકલ હશે, બધામાં એક બંડલની સેપરેટલી એન્ડોડર્મિસ, સેપરેટલી પેરીસાયકલ જે આપણને જોવા મળે છે. So the cell wall is made up of the cellulose, and the cells contain the reserve food materials like the starch grains. Starch. The cells of the ground tissue next to the hypodermis are the smaller in the size, like the polygonal in the size and the shape and the compactly arrangement. And towards the center, the cells are the very loosely arranged, rounded shape and the bigger in the size. And the vascular bundles is embedded in this tissue. મેં પહેલા જ કીધું કે આની અંદર જે આ ટિશ્યુ સિસ્ટમ હશે એ એમ્બેડેડ થયેલી છે આ પેરેન્કાઇમેટસ સેલ્સની અંદર. So the ground tissue stores food and performs a gaseous exchange. In this case, these cells are the very loosely arranged. So that's why gaseous exchange is very easily. Then the vascular bundles. So vascular bundles are the scattered in the form अलग अलग छूटा छवाया હોય છે and in the parenchymatous ground tissue એટલે જે આપણે હાઇપોડર્મિસ ની નીચેના રિજન ની અંદર આપણને જે પેરેન્કાઇમેટસ સેલ્સ જોવા મળે છે એ સેલ્સ ની અંદર જ અલગ અલગ સેક્ટર ની અંદર આ ગોઠવાયેલા હોય છે. Each vascular bundles is surrounded by sheath of the sclerenchymatous fiber and it's called bundle sheath. Bundle sheath કેને કહેવાનું સમજાણું કે each vascular bundles is surrounded by the sheath. બધા વાસ્ક્યુલર બંડલ એક એક sheath સાથે કવર થયેલા હોય છે. The vascular bundles are the conjoint type, collateral types, endarched types and the closed type. કોઈ પણ ટાઈપનું હોઈ શકે છે. Vascular bundles are the numerous, small and closely arranged in the peripheral portions. And towards the center, the bundles are the comparatively large in the size and the loosely arranged. Then the phloems. So phloem is the monocot stem that consists of the sieve tubes and the companion cells. So another parts are the absent. So phloem parenchyma and the phloem fibers. So both are the absent in the monocotyledon stem. Then the xylems is arranged in the Y-shape form. તમે જો Y shape ની અંદર જોવા મળશે. જે ઇમેજ ની અંદર પિંક કલર જોવા મળે છે, બે મોટા બે નાના, એટલે એ બધાને ભેગા કરો તો શું થાય, Y shape ની અંદર arrangement જોવા મળે. So the two metaxylem vessels are the located at the upper two arm and one or the two protoxylem vessel at the base. એટલે બે ઉપર હશે બે નીચે હશે. તો એ જે અરેન્જમેન્ટ છે એ વાય શેપની અંદર ગોઠવાયેલી હોય છે. In the mature bundles, the lowest protoxylems disintegrates and forms a cavity and known as a protoxylem lacuna. જે પ્રોટોઝાઇલમ હશે એની અંદર આપણને કેવિટી જેવું સ્ટ્રક્ચર જોવા મળે, જેને આપણે શું કહેશું, પ્રોટોઝાઇલમ લેક્યુના. Now here not a pith is distinguished. So obviously ground tissue is not differentiated, so that's why all the region is a ground tissue. So not we are the write down their part is a pith, right? So all the part is a ground tissue, and this ground tissue is made up of the parenchyma, and the vascular bundles are the embedded in the parenchyma. So the structures of the monocot stem. Now in this image we are the very closely, and in between we are the relationship told which is a differentiate in the vascular bundles arrangement. આની અંદર ક્લિયરલી આપણે જોઈ શકીએ છીએ vascular bundles arrangement is a different, some layers are arranged in the different manners, right? So what is a major difference in monocots and the dicotyledon stems? So normally in a monocot stems having the no pericycles, hypodermis is made up of the sclerenchymatous cells, ground tissue is not differentiated, starch sheath is absent, right? And vascular bundles are the closed medullary rays is absent, and the vascular bundles are the scattered in the ground tissue. So they all the characteristics of the monocots plants. Now the another is a dicot stem. So dicot stem is a hypodermis is a made up of the collenchymatous, right? And ground tissue is a differentiate into the cortex, endodermis, pericycles, and the pith. Starch sheath is a present, pith are also present. Vascular bundles are the arranged in not a scattered form, right? And the particular one manners in the arranged. So that's why the here pericycles and the piths both are the present, medullary heads are the present, and by the medullary rays, two vascular bundles are the differentiated, right? And the vascular bundles are the arranged in the one single manners, that means the ring arrangement are the seen. So they are the major difference between the monocots and the dicot stems, right? હવે એક પાર્ટ જે બાકી જાય છે મોનોકોટ્સ અને ડાયકોટાઇલિડોન્સ લીફ, જે આપણે નેક્સ્ટ લેક્ચરની અંદર જોશું.